biology class. I am Shipra from ADARSH MANDIR SHANKAR Mount Abu. For class 12, continue our topic molecular basis of inheritance. Today, we will learn about the properties of genetic material. Why DNA is better than RNA as a genetic material, and Manselson and Stoll experiment, and the process of DNA replication. So first, we will start with the properties of genetic material. Actually, the genetic material, it should be able to generate its replica. Replica means the duplicate copies. So DNA and RNA both are capable to replicate. Then second is chemically and structurally be stable. The genetic material, it should be chemically and structurally be stable. In this comparison, the DNA is less reactive chemically. So and structurally it is more stable than RNA. So DNA is better than the RNA. Now the third is it should provide the scope for mutation that required for the evolution. So both are capable for mutation. And the fourth property is it should be able to express itself in the form of Mendelian characters. So it is possible in both RNA and DNA. So according to these properties, the DNA is better than RNA because DNA is structurally more stable than RNA. Now we come to the DNA replication. For the DNA replication, the semi-conservative method is the best method for DNA replication. In this method, the two strands of DNA are act as a template for the synthesis of new strands of the DNA. And after completion of DNA molecule, one have the parental and one is new strand. That's why it is known as semi-conservative method. Semi means half. So semi-conservative, one strand is parental. It shows the semi-conservative. Okay, and this method is given by the Watson and Crick. And experimentally, this method is proved by the Manselson and Stoll. Manselson and Stoll did the experiment by the table. So ammonium chloride, but the ammonium chloride, it contains nitrogen 50 and nitrogen 14, two isotopes of nitrogen. So he grew the E. coli bacteria on the medium ammonium chloride. In this ammonium chloride, N15 is present. So he grew the E. coli, and by this culture medium, the two strands of DNA both are contain the N15, the heavy nitrogen, and then he grew the same E. coli on the N14 ammonium chloride. The culture medium is ammonium chloride, but it contains the nitrogen 14 isotope. And then what will be the result? The result is one strand is heavier than the other, because one strand contains N15 isotope and the other strand contains N14 isotope. Then again he grew the E. coli, the same E. coli on the medium N14 ammonium chloride. Then what happens? He produced two types of DNAs. One type of DNA contains one strand N14 and one strand N15. 114 and 115. And half DNAs contains only N14. Then how he proved that these are the N14 and N15 strands? By the centrifugation method. He centrifused in the cesium chloride. Then what happens? N15 is the heavy. So after centrifugation, it is near the base. Then after 20 minutes, by the gradation method, by the centrifugation method, this DNA contains N14 and N15. One strand with N14 and one strand with N15. So it is slightly away from the base. Then after 40 minutes by the centrifugation, he produced two types of bands. One is N14-15 near the base and one is N14-14 just away the base. So he proved that one strand is parallel and one strand is new. He proved the semi-conservative method of the DNA replication. Now this is the DNA replication method. In this is replication 4. What happens? These are the two strands of DNA. Two strands, one is 5' to 3' and other is 3' to 5'. Okay, so first helicase enzyme. Helicase enzyme activate. And what is the function of helicase? Helicase, the function of helicase is unzipping. Unzipping of both the strands of DNA. So by the working of helicase enzyme, these 3' to 5' and 5' to 3' strands are, their hydrogen bonds are breaks. Okay, hydrogen bonds, where are hydrogen bonds present? Hydrogen bonds present between nitrogenous bases. Adenine is always connected with thymine with the help of 2 hydrogen bonds, and cytosine always connected with guanine with 3 hydrogen bonds. So these hydrogen bonds will be break by the helicase enzyme and the two strands are separate. This is the first step of DNA replication. Then the two strands which are free, one is known as leading strand. Leading strand, the new continuous DNA strand is formed, which is known as leading strand. It is always in the form 5' to 3' direction. Okay, and the other is lagging strand because it forms in the segments, not a continuous. It is a discontinuous DNA segment. And what happens? The first is RNA primer is formed at the tip of 5' with the help of RNA polymerase, and it initiates the new strand. Then the new strand is continued by the DNA polymerase, and in the direction 5' to 3', a continuous new DNA is formed, and in the 3' to 5', the DNA segments are formed which are known as Okazaki segments. This is discontinuous strand, and these segments are known as Okazaki segment, and this new strand is known as lagging strand. It is a discontinuous, and by the action of DNA ligase enzyme, DNA ligase works and connect the Okazaki segments and continue the DNA strand. So this is the method of DNA replication. Thank you.